Our first presenter for the afternoon is going to be Michela Centinari, who has been one of the leading researchers on the impact of spotted lanternfly feeding on grapevine and grapevine metabolism. It's coming to us from Penn State. Michela.
Hi, everyone. Can you hear me okay? Okay, good. So hi, everyone. Thanks for the invite. It's my pleasure to share what we have learned so far on the impact of spotted lanternfly feeding on the metabolism and physiology of grapevine. So before I get started, I wanted to acknowledge our working team, which is really a multidisciplinary team, including Drew Harner and myself. So, our expertise are more on the plant side, physiology, production, but also we have experts in entomology, enology, and ag economics. And definitely also I want to acknowledge the research tech support and students who have been contributed to the project, to the work I'm going to be presenting today. And I have more acknowledgement at the end, but I wanted to show who are the main key players of this work. So stepping back, one of the many questions we have been trying to answer over the last years, probably 6 years, is what does lanternfly feeding actually do to grapevine? And here I'm specifically referring to adults, right? Adults lanternfly. And as you may know already, since this pest arrived in Southeast PA, we heard from grape growers about reduced yield, damage on fruit quality, and even in some extreme cases, decline in vine health or even vine death after these heavy infestations. But we didn't know why this is happening, how. There was no information coming from previous literature or another country about how this pest can damage the vine in such great extent. And one of the important, piece of this puzzle is when the SLF adults are in the vineyard, are infesting vineyards, which was, a little bit discussed this morning. But I want to kind of reinforce the concept that late season infestation by adult SLF are unfortunately difficult to manage, at least in our vineyards here in the East Coast. And that's for several reasons. So one, as you can see in this slide, and hopefully you can see my mouse, adult lanternfly typically show up late August. We see peak of infestation in September and even in October. And that is when most of our grape variety, the wine grape varieties, are ripening, right? And when growers are harvesting grapes, September and October. So definitely there are only limited type of insecticide that can be used, mostly, contact or insecticide with a very short residual activity. And as also someone mentioned this morning, unfortunately, growers see then reinfestation coming from the surrounding area. So killing lanternfly is not too hard. Unfortunately, growers see reinfestation happening shortly after they are controlled. Other reasons, is that it's a very busy time of the year. Growers are very busy harvesting, and then having an additional things to do, get into this to the tractor and go and spray insecticide is very time consuming. And there is a weather too, specifically for the later, I would say, grape ripening cultivar. The one we harvest at end of September and October, that is where typically we start to see more extended rain events. So even the timely management is even more complicated when you put the bad weather on top of other things. So to understand why those late season feeding can be problematic for the vines, and we already said they're hard to manage, and why they're going to be problematic for the vines, I just want to very briefly review how sugars move through the plants. We all know that leaves can produce sugar through photosynthesis, and those sugars are distributed, allocated in different plant tissues. And here I want to highlight the most important things, especially late in the season when adult SLF are in vineyard. So definitely we have fruit, right? So we need the sugar to go into the fruit for berry growth, cluster growth, and ripening. But also late in the season, a good amount of sugars is going to go to perennial tissues. And by perennial tissues, I mean canes, but also trunk, and most importantly, roots. And here, they're going to be accumulated as starch. So starches are, sugar reserve. We like to call them store energy, and it's what the plants need, the vines, especially being perennial crops, what they need in the following season to grow and to produce, a good crop, right? So they need this energy stored in year one to be productive and grow well in year two, and so on. So what happens, when you put a large number of lanternfly on those vines, right? So we know those are sap-feeder. They're pretty large insect, and as they feed on the vine and tap on the phloem, they can ingest a large amount of sap, which contains nutrients, but also sugars, right? And mostly the soluble sugar that are very important, again, for fruit ripening, production, and vine health. So we did several field trials in the quarantine zone to answer or to better understand what measurable effect are we going to see from this sap-feeding of SLF, how those effects are going to affect the grapevine health production, and how this information can then help to come up with more integrated pest management practices. So I'm not going to go to the details, of our field trials. I just want to mention that the work we do, we use control SLF infestations, which means that we enclose vines inside those homemade, custom-made frames, netting frames, and we expose the vines to known and different number of lanternflies. So we go from 0 to 1, 2, and so on, and to very, very high number. So you will see also in the next slides that I typically show the number of SLF as insect per shoot, because vines may have different number of shoots, even though we try to standardize the number. But overall, imagine that when I say 1 lanternfly per shoot means maybe around 15 or 20, 15 per shoot will be up to 200. So very, very large number. And here I want to show you a little visual aid on, how the vines were infested with different number of lanternflies. Of course, the insect is inside the cage, not outside, but we went from low number to very high. And today I'm only going to talk about the adults, okay? And those infestations were done late August until mid-October. Okay, so just want to mention briefly that we did, again, a few field trials on different wine grapes variety. And at the beginning, the first time, our first study, we were able to, I would say, mimic what growers experience in the field. So basically what we did, we introduced lanternfly inside the cages for about 4 or 5 days. And then we removed them by hand. So it was a lot of work. And after a few days, we put a new population inside the cages. So we tried to mimic what growers were experiencing by, infestation, then they kill them with insecticide, but then they have another reinfestation. And we did that multiple times and we reached about 4 weeks of total SLF exposure. Then we moved to another field trial. We expand to more grapevines, more variety. We had, I think, over 100 vines enclosed on those cages. So we were not able to do these, cycle infestations. So we let the lanternfly on the vines for, for many days, over 40, 50 days. And we did that for 3 years, consecutive years. So this is more like an unmanaged scenario, like what happens if you don't manage lanternfly population in your vineyard. Okay, so now I'm going to show you some of the main results that we found, in general, overall, across those different field study and different varieties, starting with harvest on year 1. Okay, so we infested those vines, again, late August, and then a harvest, we collect number of cluster per vine, cluster weight, etc. So this is a general trend that we saw on all our experiments with control infestation. We did not find any effect, negative effect, relevant effect on yield in year 1. Of course, the number of clusters per vine, this is set the year before. So if you infest the vines with lanternfly in August, the number of clusters are not going to change, right? But we didn't even see difference on cluster weight. But we did, unfortunately, found other negative effect, and specifically on fruit chemistry, with one single season of infestation. And mostly what we found that the total soluble sugar, which we measure as Brix, decreased with increased number of lanternfly. And also this effect, of course, intuitively was stronger with longer exposure. And I just want to show you an example of, of the magnitude of effect that we saw on our early harvest variety, Chardonnay, and a late harvest variety, which was Cab Franc. So those two, vineyard blocks are in the same locations. This is the same vintage, and those vines, just a note, are pretty old. They're about 25-year-old. So how you can read those graphs, just to guide you through how to interpret them. Each of these dots represent one vine that I showed you earlier, inside the cage, infested with different number of lanternfly. So you go from 0 up to up here, 15, which will be almost 200 per vine, okay? And the same for Cab Franc. So Chardonnay grapes were exposed to SLF feeding for about 2-3 weeks. And we saw definitely a pretty linear decrease in Brix with increasing infestation. We went from about 20 down to 15 Brix, which is very low, 15. This effect was stronger on Cab Franc, and our explanation is also because the lanternfly were on those vines for 30 more days, right? We harvest Cab Franc on September, at the end of September. The difference we found on the fruit on year 1 were not only about the Brix, the sugar content, but was also about color, specifically again when vines were infested with a very, very high number of lanternfly. And I think this picture gives you a good visual about the difference in color from grapes harvest, from vine with no lanternfly and a vine with high number. So then our students, under the guidance of Misha Kwasniewski, they made microfermentation, so very small amount of wine from each experimental vine, and they measured like wine color, total phenolics, and other secondary metabolites, that are important for wine color, but also other, wine traits like astringency. And also for the wine, I'm not going to show you the graph, but we did see, not surprisingly, based on the color of the grapes, and a decrease in wine color and total phenolics concentration. And we are now looking at volatile compounds, like how this SLF infestation is affecting aroma and flavor of wine. So this is still, a work in progress. We have a lot of data to analyze. We have 3 vintages, multiple variety. I just want to show you a preliminary result from an earlier harvest. These are our first experiment. It was done on Riesling. And so the graph shows the same trend, right? We have from vines with no lanternfly with high number, and you can see a reduction in this aroma compound, which I'm not going to try to pronounce. It's very hard to pronounce, but this compound is related to a fruity and floral wine aroma. But, I also want to mention that those numerical differences may not be big, right? You see a trend, but there are no, the numerical difference per se are not big. And also we don't even know if those different concentration will have an impact on the sensory perception of the wine. So we really need to do more work. I just want to show you this as an example, because from what we have seen so far, it seems like compounds, like, this aroma compound that are affected by fermentation kinetics and activity are, and most importantly, by the sugar content of the grapes, are going to be impacted. So basically, we see lower Brix sugar content at harvest that affect fermentation activity and that may likely affect those aroma compounds. So it's kind of an indirect effect due to the fruit composition at harvest. So, of course, grapes are perennial crops. So after harvest, we did measure other parameters. Starting from in the fall, we did measure or collect roots from all these vines, and we measured the starch, right? As I mentioned earlier, this for us is a good indication of the, I would say, overall health of the vine, if the vines have enough energy to grow in the following year and to produce, a nice crop. So again, unfortunately, we found a pretty strong effect of prolonged exposure. So here we're talking about, over 40 days of SLF infestation on starch accumulation in the root system. So these are similar, graphs, same vineyards, same cultivars, and just show how the starch in the root system, in the woody roots, are really declining pretty sharply. We go from like something like 20% to 5 or 2% in Cab Franc, and if you look at these two trends, they're pretty similar. I mean, they're very parallel, okay? And so it seems like to be affecting both cultivar the same way. So those numbers, doesn't tell you much, right? But when we reach value of 2, 4, 5%, those, are kind of very low starch value. We talk about carbon or starch starvation. Okay, and so we did expect to see potentially a carryover effect in the following year. So this a summary also what we saw in the following year at harvest for the same experiment. So in year 2, we actually start to see pretty strong reduction in yield, depending, of course, on the density and on the exposure. But we were not, surprised. This likely is a combination of carryover effect from the previous year, but also accumulative, like we reinfested the vines with Lanternfly. So these are repeated infestation. And I want just to show you, a summary of our yield result from the Cab Franc 3-year of data collection. This table was made by Flora Acevedo, and I think show very well the magnitude of the yield reduction that we saw. Of course, those are, control infestation. Vines are exposed to Lanternfly for a, extended amount of time, but it shows us, the yield was reduced. This is a percentage reduction compared to our control. Control means healthy vines with no Lanternfly. And you can see when you have about, 20, 23, our yield reduction were around 4 or 5%. They are stronger reduction with increasing number, and even, going from year 2 to year 3 with this, cumulative infestation effect are even stronger over time. And, we did some of our vines also decline in health following, this heavy SLF pressure. So the next question was, what about, the young vines? We saw this strong effect on mature field-grown vines, but we also know the carbohydrate, those sugars are very, very important in young vines, as during the vineyard establishment. And we know that the vines, this is the time the vines are building like a root system and a trunk, so the permanent structure, and they need a good amount of carbohydrates. So to answer, this question, we then set up another experiment with 3-year-old vines, still Cab Franc, and we had this was done in the greenhouse in a quarantine facility, and we split our vines in 2 groups. So we had, vines with no Lanternfly, and another, half of the vine they were infested with pretty high density. Those vines had, about 3-4 shoots, and we exposed them to about 40-60 SLF per vine for over a month, around a month. So what we did, since those are potted vines, we could deconstruct, and do destructive measurements of the biomass. And what we found, hopefully that's clear from the picture on your left, that the vines with this high number of Lanternfly had a smaller root system than the vines with no Lanternfly. And you can see also here on the graph on your right what the quantitative difference was between these two treatments. But definitely was visual also, was when we washed the roots, we could clearly see that those, vines produce different root biomass. We were a bit surprised because the infestation were done, later in the season. So we weren't expecting such a, significant effect on the biomass at that point in the season. Of course, this experiment was done in pots, and we need to see what's going to happen in the field, right, where roots can grow more without the constraints of a pot. But it's hard to do in the field. But we did other potted trials with young vines, and we saw some similar results. Okay. Then I also want to mention that we measure again starch and soluble sugars at the end of the season, both on the roots of those vines, but also on the canes or shoots at that point, canes, to see, if there was also an effect on the above ground perennial tissues, as those canes are very important and will likely be the next, cordons of the vine. And unfortunately, both below ground and above ground tissue were negatively affected by SLF infestation. Both canes and roots had lower starch, mostly starch, at the end of the season. Okay, but again, those vines were infested for about a month, and hopefully this is not something you're going to experience in your vineyard. So what does it mean for management, right? So we always tell growers SLF is a stressor, is an additional stress that the vines are experiencing. It's very important, and I know it's easy for me to say, but hard for a grower to do sometimes, to prevent this prolonged late season exposure. It's very important to be on top of the management, because otherwise, there can be negative effect, not just on that year, right, on the fruit and fruit chemistry, but potentially also on the next season growth and yield. Of course, repeated infestation year after year can increase those risks. And young vines, I don't know if vulnerable is the most accurate term, but definitely you need to pay extreme attention to young vines, because you want to establish, strong young vines that hopefully will be in production for 20, 30 years. And so they need, good root system and permanent structure. Then, of course, the impact will depend on many factors. But I want to highlight also the vine health point, because I show you data from our experiments. Vines have different age, but also, results can be different if you have other issues in your vineyard. What if there was a drought, or vines have a downy mildew, or other, issues that make them more vulnerable and more, less healthy, and more are at risk of high damage to the lanternfly. So next, was checking how I'm doing on timing. I have just another few slides to show what basically we want to do, I guess, next. Because from a management perspective, the practical question is, how long can vines tolerate SLF before we start to see this measurable damage? Now, what I show you mostly is like what happened after 4 weeks, or even, longer than 4 weeks. But how long does it take to see an effect? Just a few days, a week, 2 weeks? So this is going to be very useful information that growers really need in terms of managing effectively this pest. So I'm going to show you just a couple of data. These are very preliminary data that we collected from our previous experiment, but really we need to do more systematic work on this topic. So for some of our experiments, we were able to collect, for example, berries during the infestation, not just at the end, but during the SLF exposure. And for example, we measured berry weight, which gave us an indication of how much sugar, carbon, was allocated for fruit growth. And again, this is just a snapshot of 1 year and 1 cultivar. Briefly, we found that when we were into 12 days into the exposure, SLF exposure, we didn't really see an effect of SLF numbers or density on berry weight. But then when we collected, 20 days later, those berries from the same vines, we definitely start to see an effect, a much stronger effect. There is still some variation. We have, a vine here. This vine, I don't know what, with this vine, it always that's great, despite, high number of lanternfly. But overall, we saw a decrease, in berry weight. Similar trends we saw for bricks, and soluble sugar progression throughout the infestation. This is from another variety, another field, but we saw kind of similar trend, right? The day we exposed the vines to lanternfly, we collected the berries. Of course, there was no effect, of lanternfly on bricks, and that is the graph on your left. There is no trend. But then we collected berries from the same vine 11 days later, and we start to see a trend. There is still a lot of variation among these dots. But when we collect the sample 15 days later, then we really started to see a strong trend, more linear and a very clear decline in bricks. So again, just to show you how the duration of exposure can have a strong effect on those parameters. And I also want to briefly show you some data that we collected on SLF effect on vine water use. We know, of course, lanternfly is feeding on the phloem, but we know also that water transport and carbon or sugar transportation, they're linked processes. We know that this pest can also affect the carbon assimilation and photosynthesis by promoting the stomata closure. So we wanted to see, is this infestation also going to affect vine transpiration, vine water use? So we did that for 2 years. That was part of Drew postdoc work here at Penn State. And we put those subflow sensors on the vine, on the trunk of the vine, which to estimate, again, the amount of sap that was moved from the roots up to the canopy. And I have only one graph, and this is a pretty busy figure, so I'm just going to highlight a few important points, still into the context of how long may take for SLF to show an effect on the vine, right? So if you look at these lines, the gray one, those are control vines with no lanternfly. And if you look at the green, those are vines that were exposed to a very high number of lanternfly, about 180 insects per vine. So what we start to see, this is our subflow that we measure continuously through our experiment. What we start to see after 7 days, this is 7 days into the infestation, we start to see a decline in vine water use on the vines with high number of lanternfly. And this decline, this reduction were, of course, more pronounced and stronger with continuous exposure. I would say under our, environmental condition here in our humid Southeast PA, maybe these differences are not biological relevant. We saw a reduction in water use. We have plenty of water, but these results could be more, relevant for region with more limiting water availability. And we need to explore more about these trends. So where do we go next? I mentioned, how we want to do a more, cohesive and systematic work where we look at how timing of infestation, for example, during ripening or post-harvest, affect those parameters. For example, if you have, 1 year of infestation in your vineyard, and that happens early in the season or later in the season, pre or post-harvest, how this is going to affect the vines, how this will guide how you manage, those infestation. So there is a lot of follow-up study we want to do based on this preliminary, maybe not preliminary, multi-year work that we have been doing here at Penn State, depending on funding, right? So finger crossed. We also want to expand our work that was mostly done on vinifera. We have one study for 1 year only on a hybrid variety, but really we want to expand our work on Concord. We did receive a grant, didn't start yet. Hopefully soon we receive the funding, but was recommended for funding to expand, I guess, what we have been doing in vinifera now on Concord and see how, this infestation may affect important yield parameter, juice chemistry, and some of the flavor metabolites. We did plant, I want to show you here, some about 100 Concord vines here at our research station now that we are in the quarantine area. And we also have older Concord vine that we can use for these experiments. And with that, hopefully I can go over time. Again, I want to acknowledge also, these are just some of the people. There are many more that helped us throughout those experiments. It's a lot of work, and also to travel far away sometimes to do this experiment that were not going to be possible to with our grower cooperators and the funding sources that are listed here on the slide. With that, I'm done. Thanks for your attention, and I don't know if we will answer question now or later, if there are questions.
That was great. Thank you, Michela. Very interesting to see the work carried on year after year, looking at the vines under multiple years of feeding. We do have a couple of minutes, and there are some questions for you in the Q&A. The first one is, do you think the food chemistry issue could be an explanation of the stuck fermentation, the fructose imbalance that we heard earlier in the day from, I think it was Emily?
Yeah, those were that was a very interesting point, and we made a note with Julie to look into that. We didn't measure the single sugar, right? The glucose, fructose. We just measured the total solubles, the bricks. But we want to really analyze more in details the sugars present in the grapes at harvest to see, if that could explain those effects. Definitely, there is a strong effect. Just 1 year of infestation on the bricks level on the grapes. Yeah.
Great. Yeah, great to get this back and forth here at the summit. So the next question here is from somebody who does outreach with grape growers and kind of looking for a digest of, how would you tell farmers? Is it fair to say there'll be a decreased quality in the first few years leading to plant death? How would you communicate that?
I would communicate, that's a very good question, but I would say we saw this effect after prolonged, right? Like 4 weeks of infestation. So my recommendation will be to be on top of the management. Don't let the lanternfly feed in for so long, because if they do, there may be some effect. They may expect, maybe they need to check on their fruit quality more carefully, yeast or other parameter, because there could be some effect. Of course, you cannot measure starch in the roots, right? That is something we do in the university, here, but, to be proactive and manage this pest, because it can really affect the vine, unfortunately.
Mm-hmm. Okay, so yeah, it sounds like the answer is...
I don't know if that answer your question, but yeah.
Yeah, it's worth managing, and it does seem, Shika, in the chats that it did. All right, Michela, there's one more question from Ashley, and it's actually very interesting looking at cofactors of vine health. And if you do have a chance, I'd ask you to type an answer to that. And keep an eye on the Q&A. So thank you again, Michela. And we're going to go now to our next speaker. Laura Nixon is coming to us from the University of Maryland, where she is working in extension. Like Michela and many of our other speakers, Laura has been on the spotted lanternfly front for many years now. So I'm very excited to hear what she has to say. Laura?
Thanks, Jacob. Afternoon, everyone. So I'm going to be talking about work that I was involved with in my postdoc position at the Lesky Lab at the USDA ARS facility in West Virginia. So we were kind of looking at, we were taking a lot of the work that Michela and Drew Harner had done previously and applying that to threshold-based sprays for management in more of a kind of uncontrolled, realistic setting. We took away the cages and let things go a bit wild. So, oops, excuse me. So our key research questions for looking at threshold-based sprays for management were, how do spotted lanternfly affect wine grape vines when they're actually left untreated? So this work was actually done in our research vineyard, which means that because it wasn't a grower vineyard, we were actually able to let spotted lanternfly go on some of our vines without treating them, and basically track how that health decline happens when you don't treat for spotted lanternfly. So we're looking at quantifying the vine health effects over long-term infestations, and also tracking the grape yield over multiple years. And then part 2 of this is, can we then compare that to threshold-based spray programs to help negate some of that spotted lanternfly damage in vineyards? So we know that in our region, growers have reported really highly increased insecticide usage for spotted lanternfly, and a lot of we've talked a lot already about how badly hit those early growers were when spotted lanternfly came in and the loss of vines. So that has kind of led to a lot of the growers getting new infestations, having a bit of a knee-jerk reaction, and triggering sprays the first time they see spotted lanternfly nymphs on their vines. And now having, up to 10 to 12 insecticide applications per year, whereas previously they may only have 2 or 3 before spotted lanternfly. So what I'm talking about today, it's a long-term trial, but it is somewhat of a preliminary trial using SLF count thresholds. And we started planning this research in 2023. So we didn't have all of the information that we now have regarding how they affect vine health that we just heard about. So we based our count thresholds on the published data from Harner et al. In 2022. So we looked at that 2022 publication, and we saw that it seemed like about 5 spotted lanternfly adults per shoot is when you start to see that deterioration in vine health. So we looked at the vines that we have in our vineyard and sort of extrapolated that out and set a threshold of 80 spotted lanternfly adults per vine. So looking at our field plot for this, this is a bird's eye view of our grape blocks here. So you can see we have 12 sort of slightly separated blocks for research. And these are interspersed with Cabernet Franc and Traminette vines. So we have the W for white for Traminette and R for red for your Cabernet Franc. So this is our planting schematic. And, as it goes often with these preliminary studies, in an ideal world, we would want to randomize these blocks. However, if we're doing a spray trial, if we randomize these blocks, we would get a lot of spray drift into our untreated controls. So the way that we set this up is we had 4 of our blocks on the west side that we left completely untreated with no insecticides for spotted lanternfly. And then our 4 eastern blocks were our threshold-based spray treatment blocks. So the way this was split up is, like I said, we kind of have 4 reps here, and we went in and counted every week. We counted every vine in all of these blocks, and if any of the vines in one of the blue blocks hit 80 adult spotted lanternfly or 150 nymphs, that would trigger a insecticide spray for just that one replicated block. And just to note, these were planted in 2020, and we started this work late on in 2023, so they were 3 years old at this point. So the measurements we took in these vineyards was we did our weekly vine counts. We also counted the number of spotted lanternfly in the untreated control vines. Although we weren't going to treat those with insecticide, it's still good information to have to see what that background population is looking like on those vines. And then, as I mentioned, those treatment plots were sprayed if we had 150 nymphs or 80 adults present. We then took vine health measurements from all vines in all of those blocks. So those were in May, we took spring bud mortality data and counted the number of developing clusters per shoot. And then in June and September, which coincided with June, is generally the height of spotted lanternfly nymph numbers on the vines, and September is generally the peak of our spotted lanternfly adult numbers on the vines. So we took photosynthesis and leaf water potential measurements from all vines. And then throughout the season, we indicated on our plot map when we had death of a vine. And then at harvest, so around August, we did bricks testing. We did this study with our horticultural lab, Lisa Tang's lab at the USDA, and her lab would track the sugars in the grapes so that we could harvest at the appropriate time. Although that only worked for our first year, I'll get into that. So at harvest time, we harvested everything from all of these vines, and we took vine yield weight, counted the number of clusters per vine, and then we kept randomly selected bunches from each vine and made them into juice to do some juice quality testing. So I'm going to run through our 2024 data first, followed by 2025 data. It is worth noting that essentially what triggered us to begin this study is that in the fall of 2023, all the way throughout 2023, we were not seeing any spotted lanternfly on our research vines. And then sort of mid-fall time in 23, we suddenly had like 200 spotted lanternfly adults per vine. Pretty classic story of how those adults show up in people's vineyards. So that triggers us to split our plots into treatment and control. And in the fall of 2023, we actually did spray those treated plots, the threshold-based plots, with dinotefran for our fall application. And then we started this study. So looking at our weekly vine counts for 2024, the top graph here shows the numbers on our control vines, and this bottom graph shows the average numbers on our threshold vines. Since the line graph shows the average, I also have put in the minimum and maximum counts for each week, so that you can see that although the averages here were sometimes below the threshold, the maximum count on the vines for some of our replicates were over the threshold. So what this showed us in 2024 is in our threshold vines, we actually had, after our first week of counting nymphs, we had a ton of first instar nymphs on our vines. We had a lot of egg masses that year. And 3 out of the 4 replicates were triggered and sprayed with Actara. And then a few weeks later, all 4 of our threshold-based replicates hit threshold and were sprayed with Mustang Max. Then both the control vines and threshold vines, of course, got a hit of 7 in late June to treat for a Japanese beetle. So that wasn't a spotted lanternfly spray, but you can see after that 7 treatment, in both the control and threshold vines, that really also knocked those final late instar numbers down for a little while before we started to see the adults moving in later in July. So then we had our harvest sort of mid-August here. So we had 2 post-harvest sprays triggered in our threshold vines, and both of those were dinotefran. So those were our venom sprays there. Looking at our vine health data for this year, so looking at our leaf water potential, we actually did have significantly decreased water potential in both the Cabernet Franc and the Traminette vines. Although it was statistically significant, it is worth noting that you don't really start to get into like drought stress levels until it is below -1.5. So although there was a difference here and the control did have lower leaf water potential, it wasn't yet approaching drought stress levels. And there were no significant differences in photosynthetic assimilation that year. So looking at our harvest data from 2024, so looking at our red grapes, the Cabernet Franc first, there wasn't a significant difference in the total berry weight per vine collected. However, there were significantly fewer clusters per vine collected in those untreated control blocks that year. And then looking at the white grapevines, the Traminette, it's flip-flopped, so there were significantly lower berry weights per vine for those, but there was no significant difference in cluster numbers per vine. Although you can see that numerically there was lower harvest in all of the control blocks for both varieties. So as I said, we kept randomly selected berries from all of this harvest and analyzed the juice quality data. So we looked at the juice pH, the titratable acidities, and the YANs for both red and white grapes here. And you can see across all of these measurements, there were no significant differences, or really any differences to see at all, in juice quality between those control grapes and those threshold treated grapes. So moving into our 2025 data. So looking at our captures for 2025, in this early season, our counts. The number of spotted lanternfly we were seeing per vine in 2025 was very, very low, especially for the nymphs. We saw a little bump of first instars, and then we really were not seeing any nymphs on these vines throughout the season. And even once we moved over into adult season, we really weren't seeing high numbers till later on. So once again, we did apply 7 in, this one's slightly wrong, in late June to both the control and threshold vines. That lower one's slightly off, they were applied on the same day. And you can see those sort of a smattering of nymphs that were starting to be there on the vines, and then after that 7 application, they went down again. And we really did not have a lot of adults until September. So we hit our first threshold spray post-harvest, so we harvested again in August. And we hit that 1 threshold of venom, so we applied a post-harvest dinotefuran in mid to late September. And then you can see that even after that venom application, the adult numbers are starting to climb again. However, this study was being done at the USDA, and after late September, there was a shutdown, so the end of this study was not able to be completed for the season. So we don't unfortunately have any spotted lanternfly counts for the rest of the season, and we will obviously have missed at least 1 threshold spray here. So we may see the knock-on effects of that unmanaged period next spring, because there will be a 3rd year of this data in this coming year. So in 2025, we actually added these spring development data collection points after speaking with Drew Harner and getting a little more advice from him on how to do some of these vine health measurements. So we looked at 2 main things. We looked at the average number of developing clusters, so average number of clusters per shoot that were developing in May. And then also looked at the percentage of spring bud mortality in May. So the way we measured the spring bud mortality was essentially, we went through and basically, if you could see that the bud was starting to emerge, that was alive. And if you could see that the bud was there, but it was not developing at all, we called that, named that mortality. So looking at these for the Cabernet Franc vines, there were no significant differences in either the number of developing clusters per shoot, nor was there any differences in spring bud mortality. However, when we looked at the Traminette, we did see that there was a significantly lower number of developing clusters in those untreated control blocks compared to those that had been treated with threshold sprays the year before. However, there wasn't any significant differences in bud mortality. And then looking at the vine health data from June and September again, for all of the Cabernet Franc vines, there were no significant differences in water potential, nor were there any significant differences in the photosynthetic rate that was measured that year. In the Traminette vines, there was significantly decreased water potential in September. These were getting kind of low, but again, it wasn't starting to approach that minus 1.5 that indicates drought stress. So although there is a little bit of a difference there, there still isn't any drought stress being indicated. So looking at our harvest data from 2025, we had a little bit of a problem through the season with black rot. So we had black rot throughout all of our vines. When it first started appearing, and we sent some photos off to Mizuho and Drew to see what was happening with our clusters. And they told us it black rot. We were like, oh, maybe this could be data that we collect. Maybe there'll be more black rot in some blocks and not others. But actually, there was just black rot all the way through our vineyard. We will obviously solve this problem next year with some more targeted sprays for this disease. But that basically meant, in terms of our harvest data, the only yield we could count was the number of clusters that had developed per vine. Since we did have clusters develop, it's just that they all, succumbed to black rot, or most of them did. So when we look at the number of developed clusters per vine, you can see that there was, for both the Cabernet Franc and the Traminette, there was significant decrease in the number of clusters per vine in those untreated control blocks compared to the threshold treated blocks from throughout the year. And since we didn't get any berries, we also weren't able to do the juice quality analysis. However, after 2 years of this study, we did assess the vine mortality. So I will say at the beginning of 2025, myself and Lisa were discussing how do we know if a vine is dying? It's not really something we've had a lot of experience with. So we're like, how do we assess if a vine is dying? But as the season went on, we realized that we could do this very simple protocol of looking at the vineyards and noting down that the vine is very dead. So the vines would decline and then die very quickly. So it was a lot less complicated than we anticipated. So from this, you can see that in those threshold sprayed blocks, after 2 years of threshold sprays, which are these blue eastern blocks here, we had 2 dead vines out of 40, so we had 5% mortality in those blocks. And then in these orange blocks in the west, we had 8 dead vines, so that is a 20% mortality rate in those untreated controls. So what have we found from 2 years of this? Well, 2 full years of SLF infestation, plus that one additional full infestation in 2023, which is kind of a half-time infestation. That led to significant reduction in yield and a significant increase in vine death in those untreated vines. We've also found that years with minimal nymphal populations, like in 2025, can still have very large adult populations that can come in and still potentially be damaging. And then looking at our threshold experiment, spraying using a threshold does reduce the effects of those heavy SLF populations. However, what we still need to work on is, for this particular study, we are going to be collecting a 3rd year of data in 2027 before we end out this stage of the experiment. But, as I've shown you the 2 treatments we have, we didn't have the space to do a grower standard spray treatment to compare to the thresholds. So there has been an expanded vineyard planted in the research blocks. So going forward, we'll be able to compare the thresholds that we set at 80 adults to grower standard spray schedules, and or start to refine that number for the threshold. Maybe it needs to go up a little bit, maybe it needs to go down a little bit. So many thanks to the Lesky and Tang Labs at the USDA who did all this work. And I think I have plenty of time for questions. I didn't have enough time to fill my slot, so.
Yeah, we've got some questions for you.
Oh, great.
So there's one question in the Q&A. Would you attribute the increase of adults after treatment in the threshold plots to lanternflies coming in from other areas?
Yes. So, as with many of the vineyard sites in our region, our vineyard is surrounded by, we have apples on one side, peaches on another, but then we have a tree line close by that is full of tree of heaven and black walnut and all usual suspects. So yeah, we definitely have them moving in from unmanaged areas.
Yeah. So I've heard folks say that the population you're managing early with the nymphs is distinct from the adults. Would you say that's a fair statement?
Yeah, so the nymphs, although they can grow really far, what we've seen in these vineyard studies is that it seems like our nymphal population is very high early on. That's probably coming from all the egg masses that they're laying on the grapevines. So one thing that we didn't do in this study that growers would probably be doing is we weren't scraping our egg masses off the vines because, we kind of want the population to be there for us to be able to test this. So growers would potentially not have such a bump of early and star nymphs because a lot of growers are scraping for those egg masses as well.
Now, I've also heard that if you're managing for berry moth early in the season, you are also managing for the nymphs. Is that ring true?
Yeah, I mean, depending on what insecticides folks are putting on, very likely, yeah.
All right. Let's look at the Q&A again. We had a question, what's the mortality of vines normally in a year if you had that 20% with only threshold sprays?
So because we didn't have a grower standard, we don't exactly have that answer, but I will say we had a buffer zone in the middle that was vines that weren't technically in this experiment. I think we had like one vine die or deteriorate. I'm not sure if it fully died in that buffer zone, but we didn't have a grower standard in this. So that would be a question for folks that spend a lot more time in vineyards.
Gotcha. Yeah, if any of the previous speakers want to unmute and chime in. Also, let me correct. You had the 20% in control, not the threshold spray, correct?
Yeah, so the threshold base spray, we had a couple of vines die, which amounted to 5% mortality, which I would imagine is quite high in an established vineyard. But we had 20% death in those untreated controls, which is very high.
Okay. Thank you for that correction. All right. There's one question in the chat that I'll pitch to our whole panelist group here, is about Texas. For Texas growers, can spotted lanternfly survive at the higher temperature range? I don't know if anybody can speak to that. Can unmute yourself.
Melody Kina has done some research. She's recently retired, but she's done a lot of research on lanternfly. She worked for the U.S. Forest Service, and she's worked with Claire Rutledge in Connecticut as well. And so Melody's able to rear lanternfly very well and to be very robust under using just growth chambers. And I don't think this is published yet, but she reared them under conditions, high temperatures and humidity levels comparable to Compton, California, to simulate the Central Valley. And she even included a 40 Celsius heat wave of a number of days. And yes, they survived just fine and laid, between 2 and 4 clutches as adult females. So I would have to look exactly at what the details of the temperature parameters are for what you're talking about in southern Texas. But so far in those extremes, we've not seen anything that really precludes any regions, at least not yet, for lanternfly establishment. Oh, Stockton. Thank you, Holly. Stockton, not Compton. Stockton.
Okay. And 40 degrees is 104 Fahrenheit.
Yep.
All right. Yes, thank you, Julie. And thank you again, Laura.
Thank you.
Great presentation. All right. Our next speaker is going to be coming from Penn State University as well. That's Molly Kelly.
Right, so thank you for having me. Today I'm going to present some preliminary data that we have on making research wines that we spiked with increasing amounts of lanternfly. So just to clarify, this is different from Michela's work, where they made small scale wines from vines, from fruit from vines that were infested with lanternfly. In this case, we were just spiking them, similar to some of the studies that they've done with stink bugs and things like that. And I'll explain why we did that in a second here. Okay, so our objectives were to determine the impact of spotted lanternfly on both juice and wine chemistry and overall quality. So what we were interested in mostly was a toxic compound that's sequestered, that the lanternfly sequester from the tree of heaven mainly, that has a bitterness to it, the ilanthone. And so we were taking a look at that. That was our main objective. And then to provide wine growers with recommendations for increasing amounts of lanternfly in their must. Okay, so the story, or this paper by Song et al. In 2018, they talked about this quasinoid compound that's found in the tree of heaven, ilanthone. And so that's the main one that's sequestered from the tree of heaven by lanternflies. And so they showed in this study, and someone might have already discussed this during this summit, but they showed that it was unpalatable to birds when they were when they gave them food that had this quasinoid compound in it, again, mostly due to the bitter quality of it. Okay, so what we did, and we've done this over a period of years, is done some winemaking both with Gruner Veltliner, Lemberger, Noré, and we added lanternfly that would mimic 0, 2, 6, 16, and 32 insects per lug of fruit, and we used standard winemaking practices. Now, these lanternflies were collected in the quarantine zone, I believe in Berks County. Drew Harner did that for us. And he collected an equal number of male and female lanternflies. And you can see here that we just used a mortar and pestle and crushed them up and added them to the must and just did research winemaking. The wines were just fermented, pressed, and there was no filtration, no fining. We did adjust SO2 at the end based on pH. And we did add some yan at the beginning of fermentation to get it around to, I believe, 250 ppm. Okay, we did primary chemistries on Gruner, Lemberger, and Noré. The juice, we measured Brix pH, TA, malic, and tartaric acids. In the wines, we measured ethanol pH, TA, volatile acidity, free and total SO2, and malic acid, and we found no significant differences. So now if you recall with Michela's presentation, she did see the decrease in Brix because she was measuring the fruit from the vines infested with lanternfly. Here we're just spiking the must with it. Okay, we measured phenolic acid, or we did phenolic assays on Lemberger juice and wine, and there we measured total anthocyanins, color density, hue, total phenolics, and again, no significant differences. And recall that Michela did find some decrease in color in her studies. Okay, so in 2023, we decided to repeat the whole thing using Noré. And so again, we got some lanternfly from the quarantine zone. So most likely, if not completely likely, fed on tree of heaven, so had some ilanthone. And we divided those, crushed them, and added them to the Noré fermenting must. And of course, we had a longer contact time than we did with the Gruner wines. We thought that maybe we would see more of an impact from that. Okay, Misha Kwasniewski did some LCMS for us and measured ilanthone in the finished wine samples. So the LD50 for ilanthone is 27.3 ppm. That's 1,000 times larger than parts per billion. So down here, are the wine samples. So we're just considering this noise and that it's negligible amounts of allanthone that was in the wine samples. Okay, we looked at the volatile aroma compounds, and you can see on the right, there's a heat map with some of the volatile compounds where we did see significant differences in them in the finished wines. There's a lot of terpenes, esters, and so we did see differences. So with this heat map, darker colors are going to indicate higher relative concentrations. So you can see with 0 Lanternfly, we're seeing higher levels of compounds that appear to be decreasing as we go down. Okay, now these are the compounds that were that had significant differences, and so we also wanted to look at the odor activity value. So that's the amount of the compound that is present in the sample divided by its odor threshold. So if that's greater than 1, it's considered significant, and that you could or may be able to detect that in the wine. So if you look at some of these, with the exception of the alcohol hexanol, all of these, the esters, the acids, some of the aldehydes, the terpenes, are all decreasing with increasing amounts of lanternfly. So that's something that we have to go back and look at why that may be happening. But it's just an interesting trend. Okay, so you would think if you have significant differences between volatile aroma compounds, some of these compounds are above the odor activity value, you would think that would translate to sensory. So it took us a long time to get permission from the university to move on to doing sensory studies with this wine, because you are dealing with a potentially toxic compound with the quasinoids. And so that took some time. But like in many studies where they see significant differences in volatile aroma compounds, that did not translate into sensory. So we looked at aroma differences, appearance differences, and taste differences. Well, you can see for both aroma here, A, no significant differences. What about taste? No significant differences. But you can see here in appearance that at 6 insects per lug, there did appear to be some differences. I went back and looked at the responses from the sensory participants, and they said that the wine appeared cloudy. Okay, well, this is research wine. It's not filtered. So I'm attributing this to some sediment in the bottom of some of the bottles of wine, giving that cloudiness. So we're considering that just an anomaly. Okay, so the conclusions just from this preliminary data is that the quasinoid was found in the parts per billion levels in the study. So not anywhere near the LD50 for that, so considered safe for humans. Juice and wine chemistry showed no significant differences, and in Lemberger, the phenolic assays showed no significant differences. And in Noré, it did not impact the sensory aspects of the wine. Although some of the volatile aroma compounds showed differences, that did not translate into sensory analysis. So some future work, even though we're pretty sure that these lanternflies did feed on Tree of Heaven, what if we worked with lanternflies that are known to have fed heavily on Tree of Heaven? You know, might they have some measurable allanthone that could contribute to sensory differences? And then also lanternfly secretes honeydew, and that can accumulate molds and form sooty mold. So we couldn't find any fruit that had contamination with sooty mold, but does that contribute to any kind of sensory differences in the finished wines? So there's a lot more to look at with this. So with that, I'll take any questions that you might have.
Very interesting. Seems like good news. Don't want to speak too quickly or too loudly. There's a question: is there any data on chronic toxicity of allanthone? This is being asked in the context of lanternfly honeydew honey.
if anyone else wants to chime in, but not that I have seen. No. You mean in toxicity in what species?
I think to humans, either, and this is in large quantities or over time. I'm not sure what. Tell me again. The ilanthone is a quasinoid, or is there any accumulation that could be possible in that category? Like a
I would
cumulative
have to,
toxicity.
yeah, I have to go back and see how it's metabolized. I'm not sure if it accumulates. Julie, are you going to chime in here?
I mean, my understanding is that, I mean, because other like food products aren't made out of Ilanthus, that those levels just aren't available. And so, so I don't think that there is a literature there. It's kind of this, it's bitter, birds avoid it. Should we be concerned about it? But I don't think there's any literature, at least that I'm aware of. This is a conversation that I think we've had in general, that other folks who are looking at impacts of SLF on wine quality, potentially Gavin Sachs at Cornell. We've had conversations with him about that too, but I don't think we're aware to date of any literature there, because again, it's just not something that people do.
Thanks, Julie.
So there's another question here in the Q&A. Well, compound question. Were there any volatiles unique to SLF heavy wines?
There weren't any that were unique to the SLF heavy wines, but we did see that decrease in volatiles overall. We didn't find anything that just appeared in the 32 insects per lug.
Okay.
We're still looking at that data as well.
And then for the sensory panel, do you think the results would be different if you used a trained panel versus consumers, specifically for bitterness?
Yeah, these are the people that were asked to participate on this panel are normal wine consumers, and they do a lot of sensory studies at Penn State, and so they're pretty trained to detect bitterness. And this wasn't about quality of wine. It was a difference from control test. So is there a difference between these wines in aroma, in taste, in appearance? And the only difference was in the appearance.
All right, thank you for that. So there's a question here from Ashley. Are there differences with the fresh juice in sensory panels? Did you study that in this?
I have not. There's been some interest in that, especially with juice producers using mechanical harvesters, getting a lot of lanternfly in the processing stages. But I have not looked at juice by itself.
All right, so that's something we'll have to hold off for.
And if I could add, I think that that's something that, I mean, after we had those results about lanternfly bycatch being an issue with mechanized harvest if sprays aren't applied, that's something that we've communicated with folks about, and I think that's where we brought Gavin Sachs in, and then also Misha Kwasniewski is continuing his work. And so I think that that's become like a critical issue, and my understanding is that they're working on it, but they didn't have data ready available to present at this summit, but it's being worked on because it's really of concern.